Now, how could such results be explained? How could the presence of agglutination and the absence of agglutination and the experiment that we have just discussed be explained? You know that agglutination happens when the antibody that is complementary in structure to the antigen on the red blood cell binds to it. Let us use this information to explain the results in the first and the second cases. As you would recall, there was no agglutination in the first case when the red blood cell of group A was mixed with the serum of the same group A. The red blood cell of group A has antigen A. This is why it is called group A. Now, when we mixed the red blood cell of group A with the serum of group A2, the serum, that is the serum of group A, has anti-B. As you know, group A has antigen A but has anti-B. So, as you would observe here, this is the antigen A found on the red blood cell coming from group A mixed with the serum of group A2, which will have anti-B, as you would observe here. This antibody can't bind to antigen A since it is not complementary to it. So what is going to happen is that no agglutination occurs. In contrast to the second case. In the second case, there was agglutination when the red blood cells of group A were mixed with the serum coming from a different group, which is group B. Group A has antigen A on the red blood cell, as you would observe here. The serum coming from group B will have anti-A antibodies. Since, as you now know, group B has antigen B on the red blood cell but has anti-A in the plasma. The red blood cell with antigen A from group A is mixed with the serum from group B having anti-A antibody. This is going to lead to binding between the anti-A antibody and the antigen. This binding is going to lead to agglutination. The same reasoning is applied for cases 3 and 4, where we are mixing the red blood cell of group B with the serum of group A, a different group, and with the serum of group B, the same like that from which we took the red blood cells. The red blood cells of group B have antigen B. The serum of group A has anti-B. So the anti-B will bind to the antigen B on the red blood cell, leading to agglutination. In the last case, the serum from group B has anti-A. So the anti-A in the serum will not bind to the antigen B on the red blood cell and no agglutination happens. To understand the concept better, let us observe the blood transfusion revealed in the figure below. The figure shows the blood of the donor added with the blood of the receiver and the results of such a transfusion. The blood of a donor was taken, which is of group B. Group B means that the donor has antigen B, as you would see here. If this blood is going to be given to a receiver that is of the same blood group, that is group B, the receiver is going to have anti-A antibodies in his plasma, as you know. In this case, the anti-A antibody will not bind to the antigen A on the red blood cell of the donor. So there will be no agglutination. In contrast to the second case, where the red blood cell of the donor coming from group B is mixed with the blood of the receiver of group A. The donor is of group B. And again, that is, that person has antigen B. When this blood is given to a person of group A, the result is going to be agglutination. Group B to group A, different blood groups. Group A, being a receiver, has antibodies in his plasma which are to be considered. The antibodies here are anti-B, since we know that group A has antigen A on the red blood cell and anti-B in his plasma. The mixing between the red blood cell with antigen B and the anti-B antibody is going to lead to binding phenomena here. This is going to show as agglutination. Therefore, in a blood transfusion, the agglutinogen, that is, the antigen of the donor, should be different from the antibody, that is, the agglutinin of the receiver, or else agglutination will happen. The agglutinogen of the donor should be different from the agglutinin, the antibody of the receiver. If they are identical, they are the same, agglutination will happen. And if agglutination happens, this is going to indicate that the blood type of the donor and the receiver are incompatible. That is, they do not get along together. Here is another example to clarify the notion. Red blood cells are taken from an unknown group. These are the red blood cells. The group is unknown. It is mixed with two different serum having anti-A antibody and anti-B antibody. Let us observe the result in each case. As you would see here, in this case, the antibody have sort of cut the red blood cell. That is, the antibody is binding to the red blood cell. This binding to the red blood cell is going to cause the clumping of the red blood cell, the grouping of the red blood cell together, which is seen here in this form. As you would observe, they are forming a clump. This is agglutination. In the second case, when the red blood cells of unknown blood group were mixed with anti-B antibodies, no agglutination happened. Notice the aspect of the blood. In this case, it was clumped, where agglutination happened, and here there were no clumping, so no agglutination had happened. The red blood cells here are somehow, let me put it this way, free. That is, they did not bind to the anti-B antibody. This is going to tell you that these red blood cells do not have antigen B. If they had antigen B, binding would have occurred with the anti-B antibodies. In contrast to the case here, where a binding had happened between the anti-A antibodies and the red blood cell, this binding will signify that the red blood cell has antigen A, which is complementary to the anti-A antibodies. Therefore, since the red blood cell showed an agglutination reaction with anti-A antibody, but didn't show any agglutination with anti-B antibodies, therefore the red blood cells belong to group A. Let us practice a little bit more in the following two cases. In the first case, the donor is of group B and the receiver is unknown. The result is an agglutination. Now, as we know, since the donor is of group B, he has antigen B. Now, the result shows that there is agglutination. Therefore, the receiver should have anti-B antibody. Remember, and the donor will look for the antigen, and the receiver will look for the antibody. The donor has antigen B because he is of group B. The result is agglutination. The agglutination result is going to imply that there is binding between the antibody of the receiver and the antigen of the donor, which is antigen B. For binding to happen, it means that the receiver should have anti-B antibodies. So, what blood group or blood groups has anti-B antibodies? The blood groups are group A and group O. So the receiver's blood group could be A or O. In the second case, the donor is unknown, but the receiver is of group B. The receiver is of group B, so he has anti-A antibody. Now the result here is no agglutination. This would tell you that the anti-A antibodies here did not bind to the antigen found in the donor. So the donor does not have antigen A. Now, which blood group or groups do not have antigen A? They are group B and O. Therefore, the donor's blood group must be B or O.